assay, then talk about sequence-based analysis of genetic variation using three commonly used genotyping approaches. Essentially, these are resequencing approaches, such as sequence capture and genotyping by sequencing. And then in the end of my presentation, I will talk about how to use genome-wide genotyping data for performing mostly association mapping. I decided to focus on that because many of you very soon or maybe already got new genotyping data and then planning on making genome-wide association scans for the phenotypes of interest. And then just mention a little bit about usage of genome-wide genotyping for studying for detecting genomic regions that are potentially subjected to some form of selection using population genetic approaches. So just very briefly, some wheat facts that of course everybody probably is familiar with. The basic number of chromosomes is 7. It is a recent polyploid, and which is quite important when you are thinking about the genotyping. Genome size is huge and it's full of repetitive elements, and the proportion of interchromosomally duplicated genes is also very high. So something about the origin of polyploid wheat. It's a relatively young polyploid species. Its diploid ancestors originated about 2.7 million years ago. Oh, I'm not using pointer. I think that I should. Okay, here it is. So they originated about 2.7 million years ago, and then sometime about 0.36 to 0.5 million years ago, first polymerization event happened that resulted in origin of tetraploid form of wheat, and it's commonly known Triticum durum. Durum wheat is a tetraploid wheat. And then about 8,000 years ago, a tetraploid form of wheat hybridized with the D-genome ancestor with Aegilops tauschayi. And that resulted in origin of hexaploid form of wheat. So when you are thinking about SMP detection in polyploid system, of course you need to take into account the fact that it is polyploid and there is a high frequency of duplicated genes. And here you may see alignment of segment of a genome between two accessions of hexaploid wheat, and you may see here that a lot of mutations that you may see in this alignment, they are in fact divergent mutations that originated in the diploid ancestors of polyploid wheat. And essentially, it is wrong to call them SNPs. These are divergent mutations that differentiate duplicated genes. Whereas SNPs, these are the mutations that occurred already in polyploid wheat, and for that reason you have a polymorphism within the population. Like in this particular case, mutation happened in the A genome, of one of the accessions, that resulted in conversion of T base to A base, and this is the true polymorphic site, and that's what we're trying to detect during the analysis of genetic variation in polyploid system. So keeping that in mind, I would like to talk about the design of custom with iSelect SMP genotyping assay. And this is one of the popular platforms that that are used in many crop species now. I would like to go through the steps that were involved in design of this assay and what is the current status of development of custom wheat isolate genotyping assays. And these are the major steps that are involved in design of in any SNP discovery project. Essentially, you are starting with obtaining the reference sequence, and if you are lucky and the genome is sequenced, you may use completely sequenced genome as a reference. If you are not, as it happened with in our case with wheat, we first started with building the reference transcriptome, and we performed SNP discovery only in coding portions of the wheat genome. Then the second step involved in SNP discovery, it's sequencing of multiple number of accessions, followed by alignment of this sequence data to the reference transcriptome using various types of software. And once you generate these alignments, then you can start SNP discovery step. And during SNP discovery, you are considering multiple factors. You are looking at the coverage of variable sites. You are applying various quality filtering criteria, and then all these steps usually are experimentally validated, and based on the experimental validation, the filtering criteria always being readjusted. So in our experiment, when we finished the SNP discovery step, we end up with the SNP database of nearly 180,000 SNPs discovered in 27 wheat cultivars, and these are coding SNPs that are found in the express portion of the genome. And then this data was used for designing of free Illumina genotyping platforms. And the last development that we finalized the design of 92,000 SNP genotyping assay that is now being manufactured by the Illumina company. So, and I would like to now to describe briefly how SNP discovery was performed in the transcriptome sequence data. And as I mentioned before, wheat is a polyploid, and during SNP discovery, we need to take care of the existence of mutations that differentiate one genome from another genome. And for doing that, we first started with the building reference transcriptome, where we used nearly 7 million 454 reads accumulated for 10 wheat cultivars. And when we performed the assembly of this,
Please hold on. Looks like we lost him. He must have gotten kicked out of the room. Yep, everyone just please hang tight for a minute. It looks like our presenter must have lost connection to the room. Sorry about that.
okay, yeah, I'm sorry, I just lost connection. It just was disconnected. I don't know what happened. So can you hear me?
Yep.
Yeah, okay. So what we did during the assembly of this sequence data, we used very strict assembly criteria so that we are able to separate transcripts that were originating from A genome, B genome, and D genome and put them into the separate assemblies. And essentially, we very effectively diploidized genome. And as a reference sequence, we used these transcript assemblies separately. And then we, at the next step, we performed very high stringency mapping of sequence data to the reference genome, allowing less than only 1% mismatch between the mapping reads and the reference sequence. And then we used genome-specific alignments where we separated reads into genome-specific assemblies, essentially, for SNP discovery. And during SNP discovery, one of the filtering criteria that was applied, we selected only those SNPs that were covered by at least two reads. And based on our Sanger validation step, this level of coverage allowed us to reach 83% validation rate. All the data that was discovered was placed into the SNP database. This is our USDA project SNP database, and you may go there and then check what are the SNPs there, and then you may even perform a blast and then find your favorite gene. In the end, this data was, as I said, used to design iSelect SNP genotyping assays, and last year we finished designing 9,000 iSelect genotyping assay, and today I'll be talking about the results that were obtained using this assay. But currently we finished designing 92,000 SNP iSelect assay that was designed in collaboration between US, Australia, UK, Italy, and France, and currently the total number of samples that are submitted for genotyping worldwide is more than 46,000 wheat accessions, and then they will be genotyped during this spring, summer, and fall of 2012. All the data that is generated or will be generated will be accessible through the project website, and then eventually everything will be migrating to the TCAP Trititsi Toolbox database. So you should be expecting within this year more and more data being deposited there. So okay, the 9,000 iSelect SNP genotyping assay. So the iSelect platform can hold up to 1 million SNPs, the bit types, and this is the annotation that we have done with the 9,000 SNPs, and you may see that we have about 2,700 non-synonymous SNPs. We have 2,500 synonymous SNPs, and the rest of the SNPs, they were either in non-coding areas or they could not be annotated. And there are some large effect SNPs that result in stop codons. This functional annotation is also quite important because it may provide you some idea about the functional importance of the SNP if you'll find significant associations. This is how iSelect genotyping assay works. Usually for each SNP, we design only one oligonucleotide probe that is then being extended extended by the DNA polymerase, and the nucleotide bases here are fluorescently labeled, and depending on which nucleotide is incorporated, you may use the fluorescence intensity for genotype calling. And the genotyping in polyploid system generates clustering that is slightly different from that you can get in diploids. And in this particular case, you may see two individuals, and this is the SNP again in the A genome of polyploid wheat. And the probe that we designed for this assay, it matches all 3 copies of genes in the wheat genome. And then after being extended, you will get the fluorescent intensities that you'll get for these 2 genotypes will look like what you can see here on the right. The dosage of B allele to the A allele will be 1 to 2 in the first case, and here you'll have only A allele, and there will be 0 of T allele. And this type of dosage, this dosage would generate clustering that where you'll have genotype, the T allele being clustered somewhere in the middle of the genotyping plot, and the separation between these 2 clusters would be will not be very visible sometimes, and that's what you may, for example, see in the data that we obtained using 9K iSelect assay. For example, you may see a very tight clustering here on this genotyping plot, and these are compressed clusters, and this data was generated by Schaumann Chow in Fargo, and you may see that the two alleles are clustering so tightly that if you would have a heterozygous individual, you may see that this differently colored data point here in the middle. It would be very hard to call heterozygous individuals in this genotype for this particular SNPs. But fortunately, we do not have very many of these SNPs on 9K iSelect assay. There are only 347. Whereas most of the SNPs, they generate the clustering is not very tight, and you may see clear cluster separation, and in some cases, like for example in the first example, you may use this data for efficient genotype calling. Okay, I see a question from Jean-Luc. I'll probably just return back. For among 46,000, I think probably about 40 to 50% are coming from the public sector, and the rest of the accessions will be coming from the private companies. And the compressed SNPs, 347 compressed SNPs, no, they are not eliminated, not all of them at least. So maybe this particular case is eliminated, but you may have compressed clusters that are separated a little bit better, and they may still be used for calling genotypes in homozygous individuals, and for that reason we still kept them. But maybe this particular example that we have here is probably removed. So if there are no any questions about the iSelect assay, I would like just to move to whole exome capture assay. So if you look at the size of the wheat genome, you may see that it mostly consists of repetitive elements, and this is just a comparison between different grass genomes, and you may see that that the fraction of unique sequences or the genes relative to the total genome size in Brachypodium is very high. Whereas if you move to wheat, then the fraction of genomic, the genic sequences relative to the total genome size is very small. And here, what we did, if you just calculate that the cost of one sequencing run on high-seq would be about $30,000, which is, of course, underestimation. It's more expensive than that. You may, by sequencing the entire genomes, you may sequence 220 accessions of Brachypodium and obtain 10x coverage for each accession, whereas you may sequence only 4 accessions of wheat and then to obtain 10x coverage. So that means that we need to come up with some kind of approach which would simplify the complexity of the genome, and then use this complexity reduced sample for obtaining sequence data. And for that, one of the approaches that we start exploring, it is called sequence capture approach, where you have a genomic sequence and then you have your gene or target gene, and then you are designing oligonucleotide probes that are complementary to your target sequence, and then you're hybridizing these capture baits, and then you pull out your targets from your genomic sequence, and thereby you can enrich the sample for the targets of interest. And for example, your target can be a gene or some kind of regulatory sequence. And here you may see a more detailed explanation how it is done in the case of SHU-Select enrichment system that is produced by Agilent company. So you may take the genomic DNA, you shear it into the small fragments, you may barcode your libraries using some kind of oligonucleotide barcoding system, and then you hybridize your you can hybridize your genomic libraries with the biotinylated RNA baits, since these baits are biotinylated, you may then use streptavidin-coated magnetic beads to pull down your targets, and then after that sequence your sample. In TCAP project, we collaborated with a group in UK, Canada, and France, and we collaborated with Nimble Gene Company to develop a whole exome capture assay for wheat. And this capture assay has already been designed, and it targets 110 megabase of exonic sequence in wheat genome. And it is the capture is will be done using liquid phase hybridization. And just first results that were obtained from the Nimble Gene Company show that the efficiency of capture, of hybridization of the probes that are targeting genic sequences in wheat genome is quite good. So these are the results of comparative genomic hybridization of 4 accessions of wheat, Rialto, Chinese Spring, Utmost, and Truman. And on the along the y-axis, you may see the hybridization intensities of each probe, and this is the hybridization intensity distribution across the target sequence. Here, in this particular case, we targeted 6,000 base pairs from the wheat genome that included genic sequence. And you may see that the coverage is quite uniform for uniform in a sense that for each of the accessions we obtained exactly the same results. And of course, these dips in the coverage, they are mostly related to the properties of the capture probes rather than to the genotype itself. So this capture assay now currently at the testing phase. We start our first capture experiments, and soon we should have better idea how efficient we are in enriching in sequencing the exome of polyploid wheat. So. And of course, just I would like to mention about that, but I'll not go very deeply into the details. So genotyping by sequencing, this is another strategy that we are exploring. Dave is asking how much of this particular context sequence is gene sequence. Frankly speaking, I do not know. But what I do know that we do probably we include on the assay we have a lot of genes that include intronic sequences, and intronic sequences can be sometimes quite long, but certainly all the repetitive elements were removed from the capture assay. So I would assume that we probably have maybe from 10 to 15% of intronic sequences still left in the capture assay. But most of the coding, most of the capture is covering coding regions of the wheat genome. So in TCAP project, we're also exploring genotyping by sequencing approach, and many of you probably have heard about that. And it is based on the restriction digest of genomic DNA with the restriction enzymes. And in our case, we are using PST1 restriction enzyme that is methylation sensitive, and then combination of paired with the other enzymes, and mostly we focus on MSP1, and this work is done in collaboration with Jesse Poland. And then all these libraries are barcoded, and then so that you can mix together from 48 to 96 samples. And then after PCR, these samples are loaded into one lane of HiSeq 2000, and then this data can be used for can be compared with the sequences of the tags. Tags can be grouped, clustered together, and then you may discover SNPs within these tags. But this technology is still being tested for various purposes, and it's more at the development phase now, and we'll have more results later. So now I would like to switch to application of high density SNP genotyping for various purposes, and of course it can be used for it. First step that we first thing that we have done, we developed SNP maps. Then it can be applied for association mapping, analysis of diversity, linkage disequilibrium, then studying population structure, and for detecting genomic regions that are subjected to selection using population genetic approaches. So in total, in a project, we using a 9K iSelect genotyping assay, we genotyped 12,000 samples. That includes various cultivars, wild and domesticated emmer, then synthetic lines, and a set of mapping populations. For validating our mapping data has been also validated using hybridization with the ditylosomic lines of wheat. So for those of you who are not familiar with what ditylosomic lines are, these are the lines where the chromosomal arms are missing because wheat can tolerate aneuploidy, and And the absence of the chromosomal arm usually result in change in the intensity of hybridization. Like, for example, this is the example of diethyl 1AS line, and you may see that the hybridization intensity for a SNP that is located on the long arm of chromosome 1A is changed in diethyl 1AS. And the diethyl 1AS line, it is the line that has the 1AS arm and the missing 1AL arm. And this data was used for assigning SNPs to the chromosomal arms. Currently, we finished generating integrated genetic map for using 9K iSelect data. Here we used a combination of several mapping populations, including synthetic Apata mapping population. Then there is a magic population which is generated by crossing 4 by 4. So there are 8 parents in this cross, and this population, which was done in collaboration with the Australian group, Matt Hayden and Colin Kavanagh, we mapped more than 6,000 SNPs, and then there was also a population that was provided by George Dukovsky, where we mapped 2,700 SNPs, and then all this data was integrated, and in total we have nearly 7,000 SNPs that are mapped in this mapping population. And just for comparison, it's a synthetic comparison of the synthetic Apata map with the magic map. You may see that they're actually producing quite collinear results, and there are not that many SNPs that are assigned ordered differently between these two mapping populations, which helped us to integrate maps together into one reference map. Then this reference map is currently being extended. We are trying to develop within TCAP a synthetic Apata reference map, which would integrate 2,700 mapped SNPs from 9K iSelect assay, and then include DART markers, SSR markers, and the GBS tags that generated for synthetic Apata mapping population. And currently in total this reference map includes more than 60,000 markers, SNP and SSR and DART markers. Now I would like to switch to association mapping. And as an example here, we used mapping population that was built using cultivars from Great Plains. These are 300 hard red winter wheat lines, and we called it hard red winter wheat association mapping panel. Previously, we investigated the population structure of this panel, and you may see that the individuals in this population, the population structure is not very strongly expressed, and it should represent not very big problem for finding correct associations. And as you probably all of you know, that population structure should be taken into account during the association mapping, because if the populations that you are investigating are differentiated in allelic frequencies, but also they have different frequencies of the causative mutation, then you will start getting a lot of associations that have nothing to do with your causative mutation. And of course this problem has been resolved, and algorithms for statistical analysis and comparison of population structure and association mapping were implemented in the TASL program, and we recommend to use it. We accumulated a lot of phenotypic data for this association mapping panel, we had nearly 6,200 SNP markers mapped in this population and 24 SSR markers that are linked with the known disease resistance genes. We investigated the extent of linkage disequilibrium in this population, and you may see that in this population we have a 50% reduction in the extent of linkage disequilibrium within 6 to 7 centimorgans. But now the next thing that we investigated, we just wanted to be sure that the population association mapping population shows variation in the phenotypic traits of interest. And for example, we investigated for stripe rust, we investigated the distribution of the frequency of different levels of phenotype for infection type, for severity, and then plant height. And you may see that we have quite a broad range of phenotypic values within population, which actually make it quite useful for mapping these traits. So this is just the summary slide that shows the clustering of phenotypic data that was accumulated for this population. But currently, this population is included into TCAP project, and it will be phenotyped for a number of phenotypic traits, including drought and disease resistance, and if I'm not wrong, for nitrogen use efficiency. But one thing that we wanted to explore, and which is quite important, and unfortunately in many studies that were published, this question was not addressed properly. It is how many markers you really need for genome-wide association mapping. You may perform association mapping using even rather limited number of markers. Even if you have a couple of hundred markers, you may detect significant associations. And but the question that but one thing that you do not know in this case is how many associations that you are missing in your population. So and for that reason, one way of assessing the coverage of your association mapping panel is to perform so-called SNP hiding simulation experiment, where you are taking SNPs from your association mapping panel, and then you use the SNP data for simulating the phenotypes. And then you are considering and then you are hiding this SNP, and then you are considering the simulated phenotypic trait instead of SNP, and then you perform association mapping, and then you are estimating how many times you will find SNPs in the neighborhood of the hidden SNP that are showing statistically significant associations. And here, this is just the description of the simulation algorithm, where you are simulating phenotype Y, and then you may assign different levels of heritability, which actually defines the kind of introduce the variation in your data, the environmental variation. So we performed this experiment, and then we tried to assess the power of our association mapping in our population, where the power is the proportion of simulations in which at least one causative locus could be detected with the false discovery rate less than 0.1. And on the top graph, we try to assess the power to detect a trait that is controlled by 1, 2, 3, and all the way up to 10 loci with the different levels of heritability. We varied heritability from 0.1 to 1, and then we tried to assess what is the power to find association with the SNP that is located within 1 centimorgan from the simulated trait. And here you may see the results. So the results actually, of course, they are consistent with the other simulation experiments. And here you may see that the trait that is controlled by one locus, this is the top graph, and you have quite a high probability of finding correct association even for the traits with the relatively low heritability level. However, when the locus is when the phenotypic trait is controlled by multiple loci, like for example, let's take by 6 loci, you may see that you have high power to detect to find marker within 1 centimorgan only for the traits that have quite a high heritability level, and in this case it would be about 0.7. But if we perform simulation and then ask another question where we say that, well, I'm fine with finding at least one marker that is located within 10 centimorgan genetic distance, then the situation changes dramatically. You may see that this is the simulation results for a trait that is controlled by 4 loci. And you may see that even for the traits with the average level of heritability like 0.4, we have actually quite high power to detect to find SNP. So, but you should I should also note that these simulations are actually relevant only to this association mapping panel. If you move to a different association mapping panel with the different patterns of linkage disequilibrium with the different frequencies of the SNPs, then the situations with you may probably obtain completely different picture. So this is relevant only to this winter-wheat association mapping panel. So we're actually quite happy with that. So there are 2 questions, and Mike is asking if 0.05 or 0.1 are more appropriate. So Q value, it's a false discovery rate, and which is telling us what is the proportion of false leads in among statistically significant features. So in other words, if I perform association mapping and then I'll find 10 significant associations with the Q value 0.1, it means that 1 out of 10 can be wrong. And I think that these odds are quite good for the Q value 0.1 is actually quite appropriate for this case. Even 0.2 probably should be fine. But I think it depends on the number of significant associations that you can detect and you are fine with. And if you just end up with the 10 significant associations, probably using 0.05 and then discovering half of them would be too strict. So I would use 0.1 and then say that, okay, I'm fine with 1 being wrong. So the 10 different curves, what are the 10 different curves on bottom graph? So these are the, so let me, okay, let me, so 4 different loci. These are the probabilities of finding finding a marker within. These are the centimorgan distances. These are the genetic intervals. I think that we scanned. So the 1 centimorgan window to 10 centimorgan window. I just need to refresh this simulation. Frankly speaking, I do not recall that. But I think that this is the window. These are the different windows that we tested from 1 to 10 centimorgan window. Okay, so I'll just keep moving, and then if the questions will be coming up, I'll just the question from George, as a breeder investing in association, I would probably focus on a trade with the FDR 0.05 or 0.05, so that it would be nice to know the specific FDR of each of the significantly associated S&P to prioritize them in breeding. Yeah, so I think that it's just Q value would tell you about your experiment-wise the error rate, having, of course, stricter criteria is fine, but the question is just how many associations you end up in the end. And then it could be that you end up with a few associations. Let's say you end up with the 5 associations, and then if you apply 0.05 false discovery rate, then well, you'll be, of course, more certain that you are not making mistake. But I think it's just the value that everyone should decide for themselves. I don't think that there is a strict rule on which FDR is appropriate and which is not. And just I don't know, maybe Jean-Luc can correct me if I'm wrong. So if we just took a for testing purpose, we took a very simple phenotype, pseudo-black chaff phenotype, that is very easy to score, and you may see that in the field you may easily go through the plot and then quickly accumulate phenotypic data. And then, of course, we also know that pseudo-black chaff phenotype is strongly associated with the stem rust resistant gene on chromosome 3b. And you may see that by doing this association, we found right away 5 SNPs that are closely associated with this phenotypic trait. And then if you enlarge it, so you may see that the coverage that we obtained with our SNPs, at least for this particular phenotypic trait, was quite appropriate. And then by just looking at this plot, you may just easily pick up the significant association. So, and then just in the end of my talk, I would like to talk a little bit about usage of population genetic approaches that can be applied for just exploring the distribution of genetic variation in a diverse panel of cultivars. During breeding, as breeders, we are strongly selecting for a specific set of alleles during the development of locally adapted cultivars, and if there is a strong selection on a specific phenotype that is controlled by a specific set of alleles, then apparently we somehow impact the regions of the genome that contain the gene that is controlling this trait more strongly than other parts of the genome. And then therefore the patterns of genetic variation in the areas of the genome that are subjected to selection would be different from the rest of the genome. And of course the probability of detecting significant positively selected alleles would depend on the evolutionary history, and it would be probably quite easy to detect major disease resistance alleles like those alleles that were introgressed into breeding lines from alien species species or from non-adapted material, or of course the domestication traits should be quite easily detected, or the genes that are involved in regulation of flowering. And I would like to talk just about one approach that is quite broadly used in population genetic literature, which is called population differentiation test. But we also tested other approaches, but I'll skip that. I'll skip the results and focus only on genetic differentiation test. So FST, this is the statistics that is used for assessing genetic differentiation. It is the proportion of genetic diversity due to allele frequency differences among populations. And then imagine that in the first example, imagine two populations where the red and yellow, these are the two different alleles, and the number of red and yellow dots here represent the frequency in the population, and we have two populations here. And in the first case, the genetic differentiation in the allelic frequencies is 0 because we have the same frequency of alleles in both populations. But in some cases, you may see the situation like in the second example, where the two populations are completely differentiated in allelic frequencies. Population 1 is fixed for yellow allele, whereas the population 2 is fixed for red allele. And so the cases of high FST, high differentiation, usually in population genetic literature, it is said that it is indicative of regions that are subjected to strong diversifying selection. And in the case of in the breeding programs, for example, if you are selecting in a breeding program for a specific allele, then you may reach the point where you fix entire breeding program for a specific allele that you are selecting again and again in your breeding program. So, and I think that these cases should be quite easily detected, and we tried to test for that. We performed a scan for genetic differentiation across the entire wheat genome using these statistics, and here you may see the results where we took this is the genetic differentiation test for spring wheat populations, for winter wheat populations, and at the bottom we just compared population of spring and winter wheat. And you may see that there are so there is something strange here. So the graph looks a little bit odd. Okay, so please do not pay attention to the middle graph because it is scrambled. I think that it was flipped somehow. So if you look at the bottom graph, so you may see that these peaks, the peaks that you have here on chromosome 2, or for example, especially on chromosome 5, these are the areas of the genome that are located close to the vernalization gene, or genes that are involved in flowering time regulation. And there is one locus on chromosome 7. You may see that the frequency of alleles for these genes between spring and winter wheat populations, they are quite different, and it's very easy to pick them up. But when you look at, for example, top graph, where we just compared only spring wheat populations, you may see that we have peaks on different chromosomes, and these peaks actually quite different from what you can see in comparison of spring and winter wheat populations. And this can be the alleles or regions of wheat genome that contain alleles that were specifically selected in one or another breeding program, spring wheat breeding program. So by actually doing that and selecting populations for comparison, for assessing the genetic differentiation, so you may easily pick up areas of the wheat genome where you had where the breeding program applies strong selection pressure and preferably try to fix a specific allele. And that information can combined with the mapping data can be can help to actually understand what is happening in your breeding program. So just in the end, summarizing, so we talked about community resources for genome-wide association mapping using gene-associated SNPs, which were obtained by transcriptome sequencing. And now we have nice SNP assays for high-throughput SNP genotyping in wheat. We explored the utility of 9K iSelect assay for performing genome-wide association scan and detecting associations with some agronomic traits. And in this particular population, we believe that we are close to reach the point where we are performing true genome-wide association scan. Just as one of the approaches for investigating, exploring the variation in wheat populations, so you may also apply population genetic approaches, and that can actually point at the areas of the genome that were subjected to selection and breeding programs. And just in the end, I would like to acknowledge a large group of people, and it could be that some of the people are not listed here. I apologize for that, but these are the postdocs who worked on this project. I would like to thank them, especially Shishan Wong and Saifullah Kiani and Cyril Sentenak. Then collaborators, the SNP data was generated in USDA SNP project, and these are the co-PIs of this project, and we collaborated with the Australian and the UK groups on that, and we currently using all these assays in TCAP project, so the next generation sequencing was done in integrated genomic facility, and we collaborated on association mapping with the group of Bob Bowden and then Wihao Bai. So the funding is provided by USD AFRI and the Kansas Wheat Commission. There are other funding bodies that contributed other source of funding, but just they're not included here. But I would like also to thank them. And with that, I would like to finish, and if there are any questions, I can try to answer them. Yeah, I think that I think that the PowerPoint presentation will be available, at least what Dina said, right? I also have the slides. Yeah, I do not mind sharing them. There is no problem with sharing within TCAP. Yeah. Well, okay, thank you very much. So it looks like that, yeah, thanks for joining the meeting.
Okay.
Well,
thank you, Eduard.
Yeah, okay. So if there are any questions, then you may send me email. It could be that maybe something is not clear, and I removed a lot of details, and but just within 1 hour, that's how much you can provide. Okay. So that's it, Dina.
Thanks so much. Yep, I think so. If anyone is interested in getting announcements about future presentations, I'm going to walk through really quick how to join the Plant Breeding Training Network if you're not already a member. So you can hang around for that. Otherwise, thank you for coming, and we'll hopefully see you next time.
Okay, thank you very much.
Thanks again.